At this time, we're going to take questions from the panel for Dr. Ginsberg, and we have a lot of them. So let's start with Dr. Bates, and I'm going to try and alternate between Dr.
Thank you. Thank you, Dr. Ginsberg, for raising the Echeverria studies. I noticed that the tests, the neuropsychological tests they use, are also used in the two randomized. There was a lot of overlap with the two randomized children's trials, the Testapia and the New England study. But those children trials were negative in those
right.
for those tests. Can you rationalize that?
Right.
Apparent discrepancy?
Certainly, yeah. That is a good point, because if we're worried about these low dose effects, then why were those studies negative? Well, there's the issue of the length of the window of maximal sensitivity in children being more the perinatal period, likely, rather than when the CNS is more developed, which these studies started at age 7 or 8. The follow-up period is 5 to 7 years in those studies, so the full effect of amalgam exposure on neurological systems would not be expected to be manifest at that point, although it is a very useful design to do that. Probably more follow-up would be a good thing. But bigger than that, and a bigger issue than that, is that those studies, I don't feel, adequately corrected for the amount of background exposure to methylmercury that could be coming in the diet. Certainly the full amount of, for example, the Cassiopeia study had urinary mercury data, and they did not test the correlation between urinary mercury data and their endpoints. And I've been talking to Diane Echeverria at the University of Washington. They were part of the Cassiopeia study. They have the raw data, and they are looking at those correlations now, not just for the porphyrin effects, but also for the neurological endpoints, and they're writing those results up. So I really encourage FDA to look at reanalysis of the Cassiopeia data based upon urinary mercury exposure, which is a composite of background diet as well as amalgam exposures, to see whether those correlate, to see in a peer-reviewed journal publication whether those correlations hold up from what I have a personal communication about. Same thing though with the Boston study. That they had populations that had similar amounts of dietary methylmercury exposure, the amalgam and the no amalgam group, but they didn't do that wasn't part of their continuous logistic regression model. So it wasn't properly controlled for, in my mind, the amount of background mercury exposure in the Boston study as well. And a major confounder would be, as I talked about, background exposure to methylmercury, this being layered on top of that is an additional risk.
And just add, though, nonetheless, they were randomized trials, and you expect the background to be kind of much the same.
And null to the bias.
In both the groups.
And bias to the null now.
It certainly would tend to do that, but there is still that difference in terms of the amalgams in one group and not in the other.
In the interest of allowing as many questions as possible in 10 minutes, Dr. Ashton.
Yeah, my first comment is I think it's meritorious to go back and look at the interaction between methylmercury and inorganic mercury, but it's certainly possible actually that fish-consuming populations might be protected from the effects of inorganic mercury, because as we know, there are a number of studies now that show that children with higher levels of mercury, if they consume the fish, actually they might be in the upper quartile in terms of neurocognitive function. So this is just a comment. The second, I think, is more directed at the FDA, and I don't know if we can get an answer, and that is, I'm not a risk assessor, but is there any rationale for choosing different uncertainty factors for inorganic mercury versus any other compound, or is it a standard that we use 10 or 3? I mean, how do you decide on when to use 3 and when to use 10?
He had directed that to the FDA. Someone from the FDA wanted.
And I would like to comment on the first thing about the fish and the benefits of fish consumption relative to mercury, methylmercury. So, which order do you want to cover those?
Yeah. Do you want to answer that?
Yes.
The EPA?
I can answer the questions concerning uncertainty factors.
Yeah.
And that is, there are typically 5 levels of uncertainty factors. One in interspecies, intraspecies, low L to no L, chronic to subchronic, and database insufficiency. Now, the default uncertainty factor is 10. However, there is a great deal of professional judgment involved because of all the concerns about the strengths and weaknesses of the databases, the amount of data that you have, etc. So it's not always the case that you apply a 10. And in fact, it's quite common to use something less than 10.
If I could just comment on the fish studies, and in my homework I talked about the Judy Daniels study, which actually found a beneficial effect of fish consumption on some of these neurological endpoints. The benefits of omega-3 fatty acids can sometimes override the negative effects, and it's really the balance between the drag on CNS development and the benefits. And in the studies where they've corrected for the omega-3 intake, then you more clearly see the dose response for methylmercury. That's why I highlighted the Oaken studies, which are really excellent resources for seeing the mercury alone effect on the CNS.
Okay, we have to be brief because we've got 3 minutes left of this.
I really enjoyed your scholarly approach to the homework. It's quite nice. One of our speakers in the open session said that there's no safe amount of mercury in the body. And then you made a statement that there's some effect, and then you qualified it and said there's some risk if mercury gets into the brain. Can you just elaborate a little bit more?
Sure. At low dose, and again, this brings up some of the concepts from the chapter 5 of the Science and Decisions. The concept is that at doses that we would see as below the RFC, doesn't, the word safe is not the right term. Is there a risk? What is the, it may be a risk in a level of 1 in 1,000, 1 in 100,000, 1 in 10. Right now, we're not assessing the quantitative risk below the RFC. For something like mercury, where there's very plausibly not a threshold in the CNS, every molecule that gets there might have some concrete risk. The challenge is to come up what is the probabilistic level of risk per unit of exposure above and below the RFC.
Yes.
Mike Dorset here.
Dr. Dorset.
Dr. Ginsberg, thank you very much for your nice talk. Not accepting momentarily this idea of non-threshold, but in relationship to your discussion of no threshold, how does the control responses in the various studies that are indicating effects of mercury or not, how do those control responses account for the background exposures and response that you allude to and support for the no threshold, or do they not?
Well, oftentimes the control group will have some level of effect. There may be a control rate of Alzheimer's disease or auditory loss with age, and there's going to be some noise, so the control population will have some variability. A low level of exposure, say in an epidemiology study, is also going to have some variability, and you might actually have signal-to-noise problems at low doses in some of these studies, so that there might be a slope there. The advantage of having large populations to study is that you will actually, that variability can help you see a low dose slope as you recruit in more vulnerable people. With smaller groups, the control variability and the low dose variability, you may tend to bury that low dose slope.